radiological health, we work with a lot of different groups. We work with established medical manufacturers. We have a lot of different ways that we can work collaboratively with them. We also work with some startups and clinical investigators as well, and hospitals that are sponsoring medical products. And it's really interesting to note that a lot of the companies, medical device companies, over 70% of them are less than 10 people, which is kind of a staggering statistic that not a lot of people know. So it's really important for us to get the message out to some of these other companies that may not have resources of a really large company about what FDA's expectations might be when trying to get a medical device cleared or approved. So talking about clearing or approving medical devices, we typically bucket medical devices into 3 different classes. And these classes are all based on risk. So we have your lowest level risk, class 1, and essentially the requirement for a pre-market submission with this kind of device is registration and listing. And you have to ensure that that medical device follows what we call general controls, which include things like good manufacturing practice. The moderate level of risk devices are typically considered class 2 devices, and the pre-market submission pathway for these is called a 510K. The level of evidence that's needed to get a device cleared through this pathway is substantial equivalence to a predicate device. So that's essentially what we call a Me Too class. You reference a product that is already on the market and you show how your device that you've developed is as safe or as beneficial or as effective as that device. And in addition to the general controls that would be applied to the lower risk class 1 devices, there's also special controls and potentially device-specific guidances that can be applied for devices within that class. So the highest risk class is class 3. So these devices come through the pre-market application pathway. And the level of evidence that's required here is to show safety and effectiveness, reasonable assurance of safety and effectiveness. And we want to see that assurance of safety and effectiveness with scientific evidence. So again, with the regulatory controls here, we'd want to see general controls, and there could potentially be device-specific guidances and manufacturing controls associated with this kind of device. So what we do in the Office of Science and Engineering labs within CDRH and also in other research groups within the different centers of the agency is typically termed regulatory science. And I know a lot of people are probably thinking, the FDA is doing research? I had no idea. And I'm going to be honest, I was that way about 8 years ago. No idea that FDA would engage in research activities. But the goal of the research activities that we're doing is to develop new tools, standards, and approaches to assess safety, efficacy, quality, and performance of all FDA-regulated products. So that includes both in the pre-market side, where you can look at regulatory science in clinical, preclinical spaces, and also the post-market side, where we can look at post-market monitoring or any post-market signals that may pop up for a device that is currently cleared or approved. And the regulatory science can help inform a regulatory decision. It can help the reviewers in the Office of Device Evaluation to answer questions about how safe is a medical device or how effective is it. So just to give you a better idea about how our Office of Science and Engineering labs interacts with the other offices underneath the umbrella of the Center for Devices, we've come up with this little animation for you. So our office, OCEL, will generate some laboratory research, and based on that laboratory research, we can come up with new scientific knowledge. And we do publish the findings of our studies and any of the research that we do in the same medical journals, same general journals that academics would publish in, or people here would publish in. We also attend conferences and present our research to the communities that would be most interested in them. We can also use the research that we've been doing to inform guidance documents that will be used both internally by FDA reviewers and also externally to increase the transparency to sponsors or people that are coming to the FDA with applications as to what we expect from them. And some of the work that we do can also contribute to consensus standards. So all of that information can get funneled down to the sponsors, as I said, the people that would be submitting applications for medical devices. The sponsors can then take that information, incorporate it into their submissions for the regulatory reviewers to see. And we have an arrow going back and forth between FDA guidance and the review aspect of it, because the information that we bring to the reviewers is helpful, but they're also feeding us information back to ensure that the research that we are doing is going to help them out in the future. It's going to fill a gap that they've identified in some of the reviews for a particular product. OCEL personnel also act as subject matter experts for a number of different fields. We have human factors reviewers, we have electromagnetic compatibility experts, biocompatibility, animal study experts. So anytime a submission from a company would come into the FDA and there were some facets of that application that the lead reviewer or the person responsible for reviewing that file didn't understand, they would consult out to some of the personnel in OCEL to get their opinion on how a particular test was done or whether or not the data are presented in an appropriate way or were analyzed appropriately. So on the post-market side, we can also act as subject matter experts. I had mentioned earlier this idea of post-market signals. So that's when there's a product that's out on the market and there may be some sort of issue related to the use of that product. We can assemble a team to kind of figure out what that problem was and how we can modify review in the future so that that kind of problem doesn't happen again. People in the Office of Science and Engineering labs will get brought in as a part of that team to help inform regulatory review in the future. So the division that Heather, James, and myself are in within the Office of Science and Engineering labs is the Division of Biomedical Physics. You can see we are a pretty broad, we have pretty broad research topics within this division. But the majority of the folks in our division are from engineering backgrounds. But we also have people from physics backgrounds, neuroscience, biology. You know, it takes a whole team, a multidisciplinary team, to attack some of the problems that we're trying to solve. And I think our division really represents that very well. So the main research areas that we're focused in is neuroscience and biomechanics. So I was just going to talk a little bit more about the research lab that Heather and I are in. So the main goal of this lab is to advance regulatory science through the study of human interaction, both with existing technology, but also, very importantly, emerging technology. So doing some forecasting to kind of see what may be coming through the FDA seeking clearance or approval and trying to get ahead of the game to best outfit either the people reviewing these kinds of devices with the tools they need to do a proper review, and also to help the people developing the technology to make sure they've created the best evidence package possible to bring forth to the FDA. And the overall goal of our lab is really to improve therapeutic, diagnostic, and rehabilitative outcomes for patients. So our lab is fairly small, but I think we have a lot of great resources to use. Jay had mentioned earlier that we do a lot in movement analysis. So we have an 8-camera Vicon motion capture system with Bonita B10 cameras. We have ground reaction force plates, recently acquired a 16-foot long pressure mapping walkway, and have a couple of EMG systems that we can use to look at biomechanics and movement analysis. We also have a couple of EEG systems as well as a few eye tracking systems that we're incorporating into some of our research studies. And a lot of what our research is focused on now is the development of clinical endpoints for upper limb prosthetic technology. So having some functional outcome measure tests that are typically used by clinicians that are rehabilitating this clinical population is really important. So we have a few of those. So jumping a little bit more into that topic, I'm just going to hit on a few of the research projects going on in our group and then toss it over to Heather and James. But one of the things, as I mentioned, is developing and validating assessments of upper limb performance, with our main focus being on upper limb prosthetic technology in the beginning. So I don't know if anyone's familiar with this box and blocks test, but this is a pretty well used and validated functional outcome measure in a number of clinical populations to look at gross manual dexterity. And it requires the person to just pick up blocks and toss them over this partition as quickly as they can in 60 seconds. But when you think about how often you just pick objects up and throw them, that's not realistic of how we interact with objects on an everyday scale. So what we've done is a little modification of this to do a targeted box and blocks, where you pick up specific blocks and you actually have to place them in a controlled manner in a specific position on the opposite side. We also collaborate with a clinical group with national rehabilitation centers across the country. It's called Advanced Arm Dynamics. And they also jumped on this idea of needing more ecologically valid outcome measures and developed a couple of more realistic tasks that you would have upper limb prosthesis users perform. So the one on the left, the video isn't playing, but somebody picks up weights and places them down into a crate, and the other one is hanging a picture frame. So these are things that you can relate to. And what we do with a lot of this, one of the issues is not having a quantitative, objective way to look at function, to look at the effectiveness of some of these devices. So what our lab is also doing, using Vicon motion capture and ground reaction force plates, is to attach some quantities to these movements. But in order to do that, we really want to understand the normative kinematics that you would expect to find with some of these existing outcome measures. You know, if you're going to have motion capture in clinics and you have a set of quantitative data from a prosthesis user, you need to know what is normal and what is not. How far away from normal are they? So part of what our lab is doing is establishing those normative databases with the intent to make them publicly available once they're fully developed. Another thing that goes with motion analysis, Vicon is a very expensive system, and if this is something that we think will be beneficial in evaluating prosthetic technology or even evaluating other technologies, we need to see if there are other systems, less expensive systems, that can capture the same information without losing any of the important stuff. So we have a couple of studies going on looking into different motion analysis systems and how they compare to a gold standard. And there are a number of other additional projects that we have, a couple relating to upper limb prosthetics again, and we also are moving into the lower limb prosthetic area, focusing on osseointegration technology, which is a really exciting, novel space that we feel very lucky to be at the forefront of, with the innovators on. And a little bit of reverse total shoulder arthroplasty, you know, moving completely away from prosthetic technology. So it's just my intention to give you an idea of the kinds of things that we do in this lab and what research looks like at the FDA. So I will hand it over to Heather now to talk a little bit more about the VR and AR projects that are going on in the lab.
Hi, I'm Heather Benz. You heard that I'm a biomedical engineer at the Office of Science and Engineering Labs, and I'm a little too short for your mic. Okay, there we go. So I am just going to tell you a story about how we found out a couple years ago that virtual reality, as implemented in research labs today, is just really, really far away from clinical implementation. And when I say really far away, I don't mean, in a time sense. I mean just there's a lot of questions we needed answered and a lot of issues we needed addressed in order to feel that we could use it productively in our research. Oh, but I think I have to tell it from over here.
I think you have to hit the laptop.
All right. Now you can only see the top of my head, but I think you can hear me. So Kim talked about our involvement with upper limb prosthetics and novel technologies. And there have been, there's been tremendous investment in this area, which is why we're even here. There's only a handful of upper limb amputees in the United States, but there's tons of defense dollars going to it because they want to develop Luke Skywalker's hand. And we need to take something like I'm developing Luke Skywalker's hand and translate that into, okay, how do you measure whether it's helping people and whether it's safe enough? The first way we did this was helping a consortium effort, essentially, that was developing new control and sensory feedback for these types of prosthetics. So on the left, you see an actual robot that was developed by industry. And then when that wasn't available for research groups working on the sensation and the control, another group developed a virtual reality representation so that your lab at Case Western or at Utah could develop the implanted arrays that would record signals or the implanted stimulators that would give feedback and could play with it with the amputees in the loop, controlling a device, but only in the virtual world. And what we needed to know while they were prototyping was, are there differences in how patients interact with objects in VR versus the real world? Because if they're using this to develop new systems, they want high fidelity so that they know that what they do in the virtual world, how their patients learn to interact with this thing in the virtual world, is going to translate. And where are those differences and what impact do they have on product use? We're hoping we're able to generalize this. We take this as our first test case and then figure out what kinds of considerations do we need to keep in mind when virtual or augmented realities are coming into the FDA in a variety of products. So we had an experiment where we were able to duplicate tasks in virtual reality and in the real world. You see that on the top line. And then we gave able-bodied people control of both so that we knew we had the highest degree of native control possible. And how are these tasks going to go? We looked at things like time to task completion, what the body movements were when people were doing the tasks, how they manipulated the objects, and the type of grasps they used. Here we're hypothesizing that there are going to be differences because the modeling in the virtual world doesn't match the physics of the real world yet. So here on the top, you see someone struggling to complete the virtual task. They're going to get it though. I think, yes. Every time that happens, the subject, the participant got really excited. And then at the bottom, you see the real world. You'll see her wearing a bypass prosthesis. I think this is Kim. I think you're wearing the same earrings today. So you see her using a bypass prosthesis, which allows us to equip an able-bodied person with a prosthesis. And the control is slow, but it is more confident and smooth. So what we learned at a very high level is simply that there were tasks that could be completed in the real world that people could not figure out how to do in the virtual world due to a variety of physics issues primarily, but also control and feedback. When you interact in a virtual world, you're not experiencing weight, you're not getting the same type of sensory feedback, and things are difficult. No one could figure out how to pick up a small ball, no one could figure out how to put a key into a lock and turn it. These are all things that are fairly straightforward. And then when they did interact with things in the virtual world, they used very different approaches. So here you see pinch. In the virtual world, if you're not doing great head tracking, it can be easy to occlude your line of sight. And this is a simple concept, but it had a significant impact on how people were interacting with these tasks. Okay, so this is a tiny, tiny case study in a very low-risk environment. But there are big questions that result. We need to understand how to measure and benchmark VR control fidelity, understand the impact of physics model on performance. We need to understand how relevant human factors and visual display considerations come into play, and we need to be able to do this across the spectrum from folks using VR to visualize data to folks who are doing rehab with individuals with limited visual function to surgical aids, where you want to know that what you're seeing is the truth and you're able to rely on it right now in surgery. This is an open topic that we're just beginning to explore. So I'm hoping that you all can continue to brainstorm with me on the types of things we need to consider. I'll pass it over to James for the 3D printing.
Thanks, Heather. So you can see the progression here. We have human factors as part of everybody's daily life. Even in these cases, there are prosthetic users, there are people who have different capabilities, but if you bring that into something like an OR suite, human factors of all of the equipment in the OR is very important to how you're able to do your job. And if you're doing something like augmented reality or virtual reality training, you have to know that what you're doing is both going to be real to you, so that when you learn how to do it in VR, then when you try and do it on a person, it's similar enough that you can use it. And then also, as we bring that to the physical world, when you have these 3D printed models or 3D printed guides or 3D printed implants, that you can rely on those in the same way. This is one note, and it's a little off topic, but I told somebody at one point I would put this in every presentation I give from now on, and so this is my part of the presentation, and it's going in. It's because this is a question we get all the time, and so now you can take this out to everybody else you know, that FDA does not approve or clear materials for general medical use. So if everybody says something like, oh, it's an FDA approved material, no, it is not. If they say that, they mean the only thing that we approve are food contacting materials. So like the thing that wraps the chicken in the store, that's approved by the FDA to wrap chicken, right? You know, it's not approved to put in you. And what we do, the reason we do that is because something that is good for one use might not be good for all uses. And so if we just approved it for medical use, someone would come in and say, like, I'm going to have titanium, great for orthopedic implants, great for making instruments, would not be great for making an intraocular lens, right? You know, so if we approved it, though, somebody could do that. And we don't want that, so we just leave it. We don't approve them. We use materials in a context, and that's how we do that. So bring that out to everybody else. Okay. For context on 3D printing in medicine, there's a difference between 3D printing as it's used maybe in Mayo Clinic in the hospitals here and around the country to what is used by manufacturers. In manufacturing, there are people that are printing all different kinds of implants. Some are patient-specific, which means that they conform to the patient's anatomy. Some are just general, but they have some interesting geometry to them, maybe like an intricate lattice structure to get bone ingrowth in orthopedic cases. Patient-matched surgical guides, which are better for making a surgeon better able to make a precise cut in an unusual case. And dental has a whole bunch of crowns, bridges. If you've heard of Invisalign braces, anyone? Invisalign braces? All of the molds for Invisalign braces are 3D printed, and then they're vacuum formed. So that's actually the highest volume 3D printed product in the country is Invisalign, because they do hundreds of thousands a year. But if the thing that the FDA has to think about is that it might not just be Invisalign doing it. There's a case you can look up on the internet. Somebody tried their own Invisalign, so they scanned their mouth with one of these, I don't know if it was a Kinect or another optical scanner, and then they used freeware software to just move the teeth a little bit as they did, and they printed that out on a 3D printer and used a vacuum former, a homemade vacuum former, to make their own Invisalign braces. This is not FDA approved. That's fine. So what about other things? What if you were going to make a patient monitor and have something that has a little temperature sensor, pulse sensor, put a little battery in it, bed that in epoxy, and then maybe you have somebody implant that in your arm. You know, as somebody else I talked to recently, you know, probably should have just gotten an Apple Watch and probably would have been just as good. But we have to think about this whole realm of people doing things. You know, there's a lot out there, and it's not all manufacturers. It's not all Mayo Clinic. Sometimes it's a guy in a garage that goes to a tattoo parlor and puts an Altoid box in his arm. Also not FDA approved, in case there was a question. All right. And because of that, we put out things like these technical guidances or guidances on devices. Kim mentioned that is the type of output we get from the Office of Science and Engineering. We put out one for 3D printing to help people go through that process and say, what are the important things that you should look at if you're going to 3D print in a hospital, in a manufacturer, at home, wherever you're going to do it. What are those things? What makes the software good? What makes your output of that software good? What makes your manufacturing process good? Because even if you are just at home, you're kind of a manufacturer. It might be a manufacturer of one person in doing one thing, much like if you have a coffee grinder, you know, if you have one of those coffee machines that has a grinder in it, right? You're manufacturing coffee, right? Just because you're using a machine that does it for you, you're still a manufacturer of coffee, right? And it's the same way here. You've got that whole process in your home or in your clinic or in your facility, but you're acting that way. I'm going to go through just a few things about the guidance because I know a lot of people here are interested in 3D printing, and these are things that, even though it's focused on manufacturers, everybody should really take away from this. And it doesn't have any device-specific information in it, so it's just general for 3D printing of pretty much anything. And so you want to use it as a resource in addition to other things. But one other aspect of it is don't try and use everything in it. So if you look at the guidance, it's, you know, like a dozen or 24 pages long. It's got things for metal printing, for polymer printing, for making patient-specific things, complex geometry things. If you're not doing one of those, don't pull it out and try and use everything. Because we've actually literally had people come in with the guidance made into a checklist, and like, we can't check these boxes. We're not making something patient-specific. We're like, okay, fine, don't use that side. Like, just turn the page. But, you know, so really what it boils down to is these major considerations, design considerations. If you're making patient-specific gutting guides, if you're making a virtual model, if you're making some sort of a even anatomic geometries that have some sort of modification in them, what do you need to look at to make sure that's right, and to make sure you can tell that it's right if you get there? How do you know that you're not visualizing something and then putting it out in the printer in a slightly different way, so that now you don't tell until you get to an OR, or you don't tell until the surgeon has to go back 3 times to different images. Manufacturing considerations, I talked about how you want to make sure you know what you're doing. And then testing, so how do you find out that you knew what you were doing? It goes through an entire pipeline. I'm not going to talk through each of these bullets, but this is essentially the pipeline for 3D printing. You start with imaging. That's an X-ray. Obviously, you don't 3D print from a 2D X-ray, but it's easier to put that on a slide. You have some sort of 3D image or 3D volume. You go to, it is coming. You go to a 3D model, and then you put that into some sort of a 3D machine. There's about 7 different types of 3D printers that are out there, so that's a very general description of it. But this is the process. And as you can imagine, a lot of these parts of the process have a human in the loop, as we call it, or somebody making a decision or somebody making a change, so that's an area where you need training and expertise. And other parts have a computer that is just making some kind of a translational decision. And anybody who's ever tried to open up a Mac PowerPoint on a PC knows that sometimes that translation is not perfect. And that's one company with one file format doing just a software. You know, this is talking about making something coming from 5 different companies' software to hardware to a physical object. So there's a lot of transitions in there. We don't talk about regulatory policy, cell tissue printing, or other specific things. So if you don't like regulatory policy, this is your document for you. And as Heather and Kim both said, we're in the research arm of the FDA, and there are hundreds of researchers at the FDA focused on different aspects. We've brought together a team that's cross-disciplinary. So not only do we have people from the Center for Devices, we have people from the Center for Drugs and Center for Biologics that are all looking at 3D printing, or additive manufacturing as we call it, and how it affects the the medical product space. So a lot of it is in those pipeline areas that I just showed before, these design, if you have something that's implanted, maybe fatigue characterization. But then what happens when you want to bioprint a tissue and you want to use somebody's cells and put it on a biological scaffold? What is the effect of printing a cell on the cell? What's the effect of the spatial relations you can now accrue with printing accuracy? And then we actually have quite a lot into drug formulations and drug printing. There are several companies out there. There's already one 3D printed drug that's approved on the market. So that's a kind of cool one. People don't realize that it actually happens as well. People ask, oh, when are you going to get the first 3D printed device? We're like, I don't know, 10 years ago, 15 years ago. So in drugs, also, when are you going to get the first printed drug? Like two years ago about now. But it's a very growing field because people want to do personalized formulations. They want to do polypills of otherwise unmixable drugs, which you can now separate spatially in a pill. And then also bring it much like a formulary or a pharmacy into a clinical space. And we also build tools for research or tools for metrics so that if somebody has one of these machines or somebody is using one of these processes, we can help them validate that process. And that's a big word in our area, validation, so that you know what you're making, and you know that what you're making is what you should make, and you know that what you should making is effective and safe. So those 4 different areas. We identified several research gaps. These are really big gaps, so this is like the highest level, 50,000 foot level. Critical to quality parameters being one that I think in the Center of Excellence in Regulatory Science we're looking to identify some of these kinds of areas where what makes a critical change. So if you make an error in your segmentation, which is taking that image from the volume into the physical space or to the design space, how critical is that to the end product? Or if you use old material versus new material, how critical is that? If that doesn't change your product, maybe that's not a critical to quality parameter. And so we have to find all of these metrics through our research. And as everything, we want to have a public health impact at the FDA, so we have guidances that we put out, metrics that people can use, and then also, as everybody wants, the evaluations at the FDA get faster because the more we know, the better we can do this. Just talk through that. So I'll go through 2 examples. I have a couple extras, but these are, I think, fairly good and relevant ones, but I want to have time for questions if people want. We looked at making a patient-specific cutting guide. And so this is a knee, I'm sure everybody here knows that, but this is for a total knee arthroplasty and a patient-specific guide for that. So instead of using a standard kit, which is about, I don't know, 24 tools in it, you just use these 2, there's actually 3 total, but 2 pieces, make the cuts using those pieces. We found out that just in the segmentation, I mentioned that's one of those human in the loop parts, 2% change in segmentation on the bone. Granted, as I'm sure a lot of the people who do orthoradiology know, segmentation bone is fairly consistent and fairly easy. But even in this, if you make a 2% change, you can have a 1 millimeter difference in your patient-specific cutting guide. And 1 millimeter, if you move that to an angle, actually can make quite a bit angular difference, and you don't get a functioning knee, or you get a knee that might deteriorate prematurely. The different materials all had to be validated separately because this is a horrible zoomed image, I guess, but the 2 different blue lines are representative of where the guide came out on 2 different machines using the same model. So it's a completely different location on the bone. And then as anything, it isn't just the fact that you made a patient-specific model. I'm sure, anybody here an orthopod? No? Radiology? All right. Okay. So people are. So anyway, if you're not an orthopod, you probably haven't seen using these kinds of cutting guides for these instruments. But you might notice there's a big gap here. This is supposed to be flush, so it doesn't matter how good that guide is. If the surgeon can't use it, that gets back to our functional performance, human factors. If the surgeon doesn't use it right, that's not going to be right. That cut doesn't matter how good that guide is. So all of these boil down to you did the right thing. But is it useful in the situation you're trying to use it in? This is one I know some people here do phantoms for radiological either diagnostics or actually checking the machines themselves. We use 3D printing to get better, more accurate, or more realistic phantoms to do check what they have hyperspectral imaging for hemoglobin or for blood flow, and also there's a CCD camera version of that. And not only were they able to get a very representative, this is retinal vasculature, so they took a picture of the retinal vasculature and just made one physically, which is much better than making circles, straight lines, boxes, things that computers are really good at measuring, circles, lines, and boxes. They're not really good at measuring this. But they were also able to take a mobile phone camera, and if you look at that middle picture here, when you separate out the image, that looks pretty much close to that CCD camera, which is about $10,000. So we developed a little test there, which is pretty cool. This is into the weeds on printing, so I won't go into that one because I know a lot of people here aren't the printing users. But one other thing I did want to go into is to look at things like cleaning and sterility. So we had a whole project looking at that, and there's a type of printing that uses a powder bed, which means that it's like a sandbox, and then you sinter or you solidify that powder in the sandbox. And if you're making something that's very intricate, then that sand is still in there. It's like a sandcastle, so you'd have to shake it out. And the more intricate it is, the harder it is to shake out that sand. And we found out that you could measure that fairly definitively with different CT techniques, and so we're now publishing a standard on how to do that to help people, because that's a thing people didn't do before, never mind have to measure. So we're helping to build those metrics. More about that. But we also rely heavily on collaborations. So collaborations with clinical institutions. ANSI is a standards organization. America Makes is a public-private partnership, both of which collaborate to either promote the research or to create these standards. And it's much like the Radiological Society for Radiologists or for the Association of Orthopedic Surgeons. You might notice those are the two things I know more about. So I'm sure there's one for cardiology, and I know there are. But all the other societies, they all have these collaborations to promote things and foster innovation in their spaces. And this is how we really get out into the world and do that as well. You probably know why people use standards, either clinical or otherwise, so I won't go through why they're important, but this is why they're important. And recently, I know that Mayo does a lot of 3D printing of anatomy, and we've worked collaboratively with the radiologists, with Jay and Jane here, and with other people at different institutions to try and build more consensus about what is a good practice for this kind of printing. We had a meeting in August, so if you're interested, we have that all online. There's transcripts and slideshows and webcasts, so if you have a day off, good watching there. There's some fireworks in the middle when we said some stuff that industry was like, ooh. But so this was a kind of a keystone of our ability to foster that innovation across different boundaries. Usually, FDA is in a silo over here on a hill, clinicians are over here in the valley, and then never the two shall meet. But this was a really good meeting of the minds, and it helped bring some of these real-world problems to the FDA, bring some of the regulatory framework information to the clinicians, and then talk about both of them in between. And so with that, I'll just acknowledge the different teams and collaborators we have. The same with the additive manufacturing research team, and put up some QR codes in case you want to look up our websites. But I think with that, yeah, we do. We have about 12, 13 minutes for questions. Open it to the floor.
Well, thanks. That was very insightful. Really glad you're here. Does anybody have any questions for them?
Hi, I'm Missy Morrow. I'm faculty in HSR, and my question is for Kim. My background and training was all motion analysis biomechanics, so everything felt very familiar, and I was wondering, how do you choose your priorities for this lab within the FDA in terms of when you might outsource to some academic centers, labs or specific experience in upper lip prosthetics. So what are those choices to determine what you will focus on and what sort of outcomes you want to define?
Yeah, that really comes from conversations that we have both with the people in the regulation division, but also with product developers, other people that may be developing innovative technology that we can expect to see coming through in the near future. You know, we want to be as prepared as we can for some of these emerging technologies, and especially with something as nuanced as upper limb prosthetics. Heather mentioned there's very few compared to lower limb, and I think it's some 90% of what's considered an upper limb amputee, it's partial hand. It's not even getting to a full prosthetic device. So that narrows the potential expertise in that area even more. So we don't pretend to be experts in everything. We can be experts in some things, but upper limb prosthetics and rehabilitation, we most certainly are not. So knowing that, and then knowing the expected technology that's going to come through, we knew to make this project as strong as possible and as relevant as possible, we needed to get experts from outside. We needed to get expert clinicians in on this project.
And that's why they have, you see here, there's a lot of University of Maryland, Marquette, there's Arm Dynamics, Alberta, University. So we really try and bring in that external expertise.
So when you develop that expertise and you develop these testing and publish this research, then if someone develops an upper arm prosthesis, is the goal then to provide some sort of framework to say, you have to test it using these things that we've proven to be valid?
Yeah, exactly. And that's another thing about the research that goes on in our labs. It's not product specific. It's not one company's device specific. What we're trying to develop are tools and methods that can be used across a product type. So that example is great. If a company comes in with a new technology that they're developing, we're hoping that what we've developed in the lab is going to be applicable to them. And so that's part of the challenge too, understanding what's coming and what's out there. Because we don't want to forget the technology that's out there, because it works for people. And people are making improvements on things that are already out there, and those things still need to come through the FDA. So it's taking all of those things into account.
Yeah, there was another question.
So I know there is, for example, an in silico model of diabetes that FDA will accept as ground truth for example, new therapies for diabetes. When do you see VR, 3D printed objects, you know, the same technology you're talking about, serving as a true surrogate that the FDA will accept as an endpoint for approval?
How about the beginning point?
Yeah, so there are different types of in silico modeling, and CEDAR has a fairly robust in silico modeling program, I think, which is where the diabetic therapy goes through. For, say, like a replacement for an animal trial or a replacement for a clinical trial, we're a bit off from that, you know, a bit ways off. But we do have programs to foster that program or foster that development. I think we actually specifically have an in silico animal trial program now, is it? Virtual? Yeah, virtual animal program. So we have programs to foster that development, but the number of variables that you need to overcome or to encapsulate to do that is just so large that it takes a while.
But if you had like a skin, like for example, skin printing is kind of hot right now because they have actual products that can do it. And skin companies are interested in testing their products than medical grade things and saying, oh, we tested on 3D printed skin that has been proven to be, you know, just like real skin.
Yeah.
What's the hurdle they have to come over to make that claim, similar to like hard on a chip or 3D printed liver segments?
That's a good point. I think, especially for things like the tissue on a chip, we have a similar program for that as well, right? I think we have looking at Heather because she has a better idea of some of these external programs. I think that has a little bit lower barrier to entry because if you're doing a specific thing, you have fewer variables to test to. especially for skin contact or some of those irritant kind of things. Or one of the big ones now is 3D printed liver or bioprinted liver, where they're showing analogous response from that liver tissue to an animal liver or to a human liver, saying that the big one is like NSAIDs, acetaminophen kind of toxicity. They can show that in a 12-well plate. And so when you're talking about that, I think that is closer. And we have a program for medical device MDDT, medical device development tools, which if you put something through that program, and you say, I have a 3D printed liver, or I have a tissue on a chip, something or other, this is where I've tested it. It matches some sort of animal or some sort of a human response or human study. Then it gets qualified through that program, and it can be used in other people's submissions. And I guess in that particular case, you don't forfeit IP rights or licensing, any of that. So it's not something that we would take over. It's just something that we would qualify.
There are other questions? It's a good question.
Yeah, you did mention a slide that I'm probably interested in, the printing optimization. I just wanted to know.
The one I skipped over.
Yeah.
When you said this gets too deep.
Yeah, this was like a little bit more in depth. So this was about printability of materials, because especially in biomaterial printing, where you're printing collagen or fibrin or something like that, people are doing their own bespoke development of materials, and there are hundreds of them out there, and people are doing it in droves. So finding a way to look at the material characteristics, the bulk characteristics of a material, or maybe the surface characteristics of a material, to then tell, is that printable? Is it feasible to use in certain situations? That's the focus of this research, and this particular project was looking at the viscoelastic properties of the material and how well that would allow adhesion for a layer-by-layer process. And so there are several efforts to just try and characterize, if you're developing a material, rather than having to do 150 different iterations, use a model that tells you if this is your viscosity, if this is your, or viscoelasticity, if this is some bulk property, the chances of it being printable, usable, etc. are X.
Yes.
Just as a follow-up, can we think about a system to monitor this printing? Because I suppose it's expensive and time-consuming. So if we are printing something and we have some objective to reach or some standard to follow, if we are out of a standard at any point, we stop the process, or if we are being drifted, we just come back to the system, something like that.
There's some people too doing that in the 3D printing world for manufacturing. Now that 3D printing is kind of wrapped prototyping and manufacturing, they have sensors embedded inside the printer that scan every layer. See, should we finish this build?
Correct.
You can imagine aerospace doesn't want to finish an entire build of something that's going wrong
for
a quarter of the way through.
Yeah. And then that is kind of the holy grail, because it's very difficult with some of these processes to know what is that critical to quality parameter. So if you're off by 1% on some temperature, is that going to ruin your build? If your heat dissipation in your build space has been reduced in some area, is that going to ruin your build? And so that is an area of a lot of research.
Yes.
Both in the metal printing is probably the largest because it's conjoined with aerospace, but then in some of these materials and the bioprinting especially, where your material can be so variable that you need to monitor the material properties, you need to monitor maybe cell health or viability, and how do you incorporate that also? You're not just making 5 of these and you can say, I can make 5 of these, but that you can say, I can make them in a month and they'll be the same.
And even for us to do medical non-bio printing, just for our medical 3D printing we do in the hospital, everything varies by what we're doing. So if we're a millimeter off in a large pelvic model and the acetabulum's a millimeter, nobody cares, right? If there's a subtle abnormality there. But if we're doing a really tiny wrist bone and we're creating an implantable solution and it's a millimeter off, well, that really matters. So even within the same type of printing but different anatomical region or different intended use, a millimeter, a submillimeter, a micron, all matters differently.
That gets to the minimum requirements or clinical requirements for any given specialty. So it would be variable across all of them. And so that's where...
Your conference is scheduled to end in 2 minutes.
Oh boy. Well, better wrap it up.
Thank you, Siri.
So if we don't do that as the FDA, but a clinical society could set guidelines for what is a clinically relevant feature or a clinically relevant endpoint outside of the manufacturing process.
Any other questions?
Better be short. We've got a minute and a half.
Well, thanks everybody for taking the time to come.